the central and the peripheral nervous system. The central nervous system is composed of the brain and spinal cord. And the brain in turn is composed of cerebrum, diencephalon, brainstem, and the cerebellum. The diencephalon further comprises of the thalamus and hypothalamus. The brainstem is composed of midbrain, pons, and medulla. And then the cerebellum. So now going on to the basics of how this development occurs. So first, like starting from the fertilization. So this is the ovary. So the pre-ovulatory follicle, and then the ovum ruptures into the fallopian tube. The fimbriae captures, and just a minute. And then there will be multiple sperms trying to reach the ova, and finally it gets fertilized, and then the zygote is formed through various stages, the two-cell stage, and then the morula, and then finally gets implanted in the wall of the uterus, in the endometrium. So after this, so first is the two-cell stage, and then is the four-cell stage, and then is the morula. So wherein which there will be multiple cells packed within a membrane. Multiple cells packed within a membrane. So the membrane will restrict the expansion of the thing, and then it will get, and then enters the blastocyst. So the blastocyst, it is composed of two cell layers. The outer cell mass, which is called as the trophoblast. The inner cell mass, which is blue in color, is called as the embryoblast. So our focus is mainly on the inner cell mass or the embryoblast. So this is a bilaminar disc theory, wherein which this is a bilaminar disc wherein which the inner cell mass, which is called as the embryoblast, which we saw here. So this is what we are going to discuss about. So the inner cell mass differentiate into an epiblast and hypoblast. So that is why this is called as an bilaminar disc. So the epiblast, within which is the amniotic cavity, and then is the hypoblast, within which lies the yolk sac. So there are two cavities and two cell layers. One is the epiblast, within which is the amniotic cavity, and then is the hypoblast, within which is the yolk sac. So this is another cut section which is showing the amniotic cavity within the epiblast, the yolk sac within the hypoblast. So now we'll see the cut section. We'll enter into the cut section of this embryo. So now what you will be seeing is this is the epiblast layer, and this is the hypoblast layer. So this epiblast layer, the shed off cells or the dead cells are those who have finished the multiplications, and then the dead cells and all these things will get accumulated in something called as a primitive streak. So this is called as a primitive streak, which is composed of the shed off hypoblast cells, the shed off epiblast cells, or the dead epiblast cells. So at the end, so this is the caudal end, this is the cranial end. So in the cranial end, you have the, So in the cranial end, you have the orobuccopharyngeal membrane, which is the future mouth. And in the caudal end, you have the cloaca, which is the future anus. So from here, from the caudal end, there will be something called as a primitive streak, which is predominantly composed of the shed-off cells from the epiblast. So at the end of the primitive streak lies the primitive node. So this is the primitive node. So the primitive streak ends in a primitive node towards its migration towards the cranial end. So now we have already seen about the bilaminar disc. Now we will see the trilaminar disc. This is the trilaminar disc. This is called as, in other words, this is called as the gastrulation. The formation of the three layers, that is called as the gastrulation. So initially it was a bilaminar, now we are entering into the trilaminar. So now you have, this is the primitive node and this is the primitive streak. And this is the epiblast and this is the hypoblast. Now what happens is cells from the primitive streak traverses through the epiblast and move towards the hypoblast. So this will induce the formation of endoderm, and then here lies the mesoderm, and the part of the cells from the primitive streak which is in the area of epiblast transforms into the ectoderm. So thus the three layers are formed, hence it is called as the trilaminar disc. The ectoderm, the endoderm, and the mesoderm. Ectoderm, mesoderm, and the endoderm, induced by the migration of the epiblastic cells from the primitive streak towards the hypoblast. So from the primitive node is an organized cells. So here lies the organization. So the cells from the primitive node enter into the move towards the hypoblast, traverses through the mesoderm, and move towards the cranial end. So here you can see this is the cranial end, this is the caudal end, this is the oropharyngeal membrane, this is the cloaca, and this is the primitive streak, and this is the primitive node. So from the primitive node, the cells passes through the mesoderm and move towards the oropharyngeal membrane. So this tract is called as the notochord. So this is what is called as the notochord. So here lies the notochordal cells. So here this is another cut section where you can clearly see the notochord. This is the notochord. So this is how the notochord is formed. So this is a cut section where you can see this is the primitive node from which the notochordal cells move towards the cranial end through the So this is from the primitive node through the primitive pit through which the notochord cell. This is the cranial end. This is the caudal end. Here you have the oropharyngeal membrane, and here is the mesoderm. So the cells from the primitive node, that is the notochord, move towards the cranial end through the mesodermal layer. So here you can again see this is the notochord, this is the ectoderm, this is the mesoderm, and this is the endoderm. So there are certain growth factors secreted by the notochord which will induce the formation of neural plate through the ectoderm. So the neural plate is nothing but the thickening or some changes happening in the ectoderm through various factors produced by the notochord. So wherein which the neural plate starts to develop from the various factors, especially the fibroblast growth factor and retinoic acids. So through these factors, the neural plate is induced and the neural plate elongates development of the neural groove. So this we'll see in detail now. So now the neural plate is formed. So this formation is induced by the growth factors produced by the notochord. So this is the neural plate. And what happens is the neural plate invaginates towards the notochord. So it invaginates. This results in the development of a neural groove. So this is the neural groove. And the formation of the neural groove results in the formation of the neural folds. So this is the neural folds. The ends of the neural fold is called as the neural crest. So the ends of the neural fold is called as the neural crest. So these are the neural crest. So this is the neural crest, this is the neural fold, and this is the neural groove. So again you can see the same. So the neural, this is the neural groove, neural fold, and this is the neural crest, and here is the notochord. So as a result of this invagination, finally there will be fusion of these two things. So resulting in the formation of the neural tube. So this is the neural tube, and this is the neural canal, and this is the notochord, and this is the neural crest cells. So here you can clearly see the formation of the neural tube, and then the neural crest cells and the notochord. So thus the neural tube is formed. So what happens is, now the neural tube is open. So the middle neuropores, there will be multiple neuropores. The middle neuropore initially closes. So the middle neuropore initially closes, resulting in the formation of the neural tube. and then there will be the anterior or the cranial neuropore, and posterior or the caudal neuropore. Okay. So as I said, the middle neuropore closes, resulting in the formation of the neural tube. The cranial neuropore, and that is the anterior or cranial neuropore, and the posterior or the caudal neuropore closes later on. So the cranial neuropore closes by 25th day, following which the caudal neuropore closes at day 27. So the failure of closure of the cranial neuropore results in anencephaly or hydranencephaly. The failure of the closure of the caudal neuropore results in the spinal dysraphy. So thus the neural tube is formed. So the tube, which is after the closure of the neuropore, the cranial neuropore, the neural tube is formation of the neural tube gets completed. So once the neural tube neural tube formation is complete, then there will be 3 swellings. So there are 3 swellings, which are 3 primary vesicles. 3 swellings which corresponds to 3 primary vesicles. That is the forebrain or the prosencephalon, the midbrain or the mesencephalon, the hindbrain or the rhombencephalon. So now what happens is this. Now what happens is the forebrain or the prosencephalon, it is divided into an upper part, upper 2/3, and a lower 1/3. So the lower 1/3, the proliferation or the development of the lower 1/3 is much less than the upper third. In other words, the upper 2/3 proliferates in a much faster way and an aggressive way than the lower third. And the junctional point doesn't proliferate much. So what happens is there is a better growth and proliferation of the upper 2/3 of the prosencephalon, resulting in giving an appearance of an Mickey Mouse. So this results in the development of the telencephalon. So the upper 2/3 of the prosencephalon results in the development of the telencephalon. The lower 1/3 of the prosencephalon results in the development of the diencephalon. Okay. So then the mesencephalon develops in the same way, and the rhombencephalon divides into metencephalon and myelencephalon. So the metencephalon, from the metencephalon, the pons and cerebellum develops. From the myelencephalon, the medulla develops. And then is the spinal cord. So from the telencephalon, the cerebral hemispheres develop. From the diencephalon, the thalamus develops. From the mesencephalon, the midbrain develops. So correspondingly, the lateral ventricles, which is contained within the cerebral hemispheres, develop from the telencephalon. The third ventricle, which is confined between the 2 thalamus, develops from the diencephalon. The cerebral aqueduct, which is confined within the midbrain, develops from the mesencephalon. The upper part of the 4th ventricle, which is confined within the pons, develops from the metencephalon, whereas the lower part of the 4th ventricle, which is contained within the medulla, develops from the myelencephalon. So thus the development of the brain occurs. So this is a picture where you can clearly understand how the ventricular system develops. So the lateral ventricles corresponds to the telencephalon. This is the telencephalon, and the third ventricle develops from the diencephalon. From the diencephalon, the cerebral aqueduct is seen developed from the mesencephalon. The 4th ventricle, the upper part of the 4th ventricle, develops from the metencephalon, whereas the lower part of the 4th ventricle develops from the myelencephalon. So thus the formation of the brain gets completed along with the ventricular system.